Hello, my name is Luis. I'm a physics student working in Sabakun's group, and today I will briefly share the main points of our research projects concerning carbon cycling in closed ecosystems. Nutrient cycling is the cyclic flux of nutrients among organisms within an ecosystem. Life on the planet depends crucially on cycling of nutrients, and for these reasons, one can find nutrient cycles in ecosystems at all scales. For example, a large portion of the global vital flux is sustained by nutricycles. Similarly, a study of microbial communities in aquifers showed that nutrient cycles emerge to overcome local resource limitations. The research question we wanted to address with this project is, how do ecosystems self-organize to cycle nutrients? To answer this question, we decided to use closed microbial ecosystems as experimental models of nutrient cycling. Here we see an example of a commercially available closed ecosystem. You can see some producers and some consumers, which are initially provided with a supply of nutrients. And after closure, it is able to sustain itself purely by providing light, which can only occur if nutrient cycles emerge to replenish resource limitations. It is the fact that the community can only sustain itself if stable nutrient cycles emerge and persist that makes closed ecosystem promising models of nutrient cycling. Consider this diagram of a microbial closed ecosystem. Here we can see the cycling of carbon from organic to inorganic forms mediated by photosynthesis and respiration. We use the fact that in temperature-controlled closed ecosystems, carbon cycling results in measurable changes in headspace pressure. To measure these pressure changes, we use pressure sensors for cell phones, which are cheap, small, and sensitive. Each closed ecosystem is housed in a monitoring device, and the community is exposed to light and dark 12-hour phases. To the right, we see an example of data, its pressure as a function of time, and we observe that stable pressure oscillations within days as a response to the alternating light and dark phases. We performed some calculations to show that the rate of carbon cycling is proportional to changes in moles of oxygen, which in turn is proportional to the amplitude of the pressure oscillations. Carbon cycling can be measured for closed ecosystems in real time by measuring pressure. We assembled our closed ecosystems using a top-down approach. We took soil samples and using drugs isolated a bacterial community of heterotrophs. Then we added a lab strain of a well-known model phototroph, Chlamydomonas enhardi. The communities were placed in the monitoring devices and we recorded the carbon cycling rates. After some time, we took a sample from each closed ecosystem and enriched into fresh media and restarted the community, again allowing the community to stabilize and record its carbon cycling rates. We repeated these steps 3 more times. The idea is that each enrichment round selects in favor of key taxa for carbon cycling. Since the composition of the community is progressively changing after each enrichment, we can see how community structure impacts carbon cycling. To quantify changes in community structure and metabolism, we use the remaining material of each closed ecosystem to characterize taxonomic and metabolic properties. Taxonomic characterization was performed via 16x applicon sequencing, and metabolic characterization via ecoplates, which is just a commercially available carbon utilization assay. Our experiment suggested that carbon cycling emerges and persists in functionally redundant closed ecosystem. This conclusion is based on 4 key observations. First, closed ecosystem robustly self-organize to cycle carbon. Second, self-organized community structure is not unique. In fact, for increasing rounds of enrichment, we observe closed ecosystem taxonomy divergence of community structure. Third, the cycling capability seemed unaffected by these taxonomic differences. We found that all instantiations of the closed ecosystem had similar carbon cycling rates. Lastly, we show that the heterotrophic communities had similar conserved metabolic capabilities. In the future, we want to study further how functional redundancy emerges in our closed ecosystem and which, if any, benefits it confers to the closed ecosystems. I would like to thank Sepakun, Kamodi, and Session for their valuable help in this project, and I'm looking forward to the discussions on Saturday.